Hello everyone. Today we are going to study different things like genome, genomics, proteomics, bioinformatics, high throughput technology. Okay, so different things we will study. Let us study what is genome. Okay, so what is genome? See, whatever DNA is inside an organism, all that is genome. So if I say the entire library of genetic instructions, the entire library of genetic instructions that an organism inherits, right? This is called genome. A particular species has particular genome, right? Okay, now if I say, for example, humans, whatever DNA is inside you, all that is genome of human, right? Human species. Okay, Homo sapiens. Now, what is genomics? Simply study of genome is genomics. So, study of whole set of DNA. Whole set of DNA. This will have both coding and non-coding regions. So, it will have genes plus other DNA, that is non-coding DNA, also coding DNA, that is genes are only 1 to 2% of your genome. The rest is non-coding DNA, and the study of all this is genomics. Now what we will, next thing is there, genome sequencing. See, you know your DNA is a sequence of nucleotides, and studying the entire sequence of nucleotides for a representative member of species. Representative, for example, a particular human being, for a representative member of a species. So simply entire sequence of nucleotides, the A, T, G, C, right? So that entire sequence is genome sequence. We have, if I say in your cell, okay, one cell, there are two similar sets of chromosomes, right? And each set of chromosome, if I say for human, you have two sets of chromosomes here. You have 46 chromosomes, right? Two sets of chromosomes, and in one set, right, there are 3 billion nucleotides. Okay, nucleotide pairs of DNA. So 3 billion nucleotide pairs. Clear? Okay. The genome sequencing has been done for many organisms, for many animals. Humans are animals, and there are other animals also. For them, for humans, for different plants, for different bacteria, for different fungi, bacteria, archaebacteria, right? Archaea. So for different organisms, the genome sequencing has been done. Okay, so for this you need, if you have to study the sequence inside a cell, right, genome sequence, you need to apply which approach? Systems biology. I have already told you earlier that we take a particular system, biological system, for example a cell, an organism, or you go to population level or any level or biosphere level. Any system can be considered, anything can be considered as a system in biology. Now here we have to go to the cellular level And in fact, inside the cell we have to go to molecular levels. For example, we are talking about which level here, which molecule? DNA. Okay, now I will tell you another thing, proteome. What is proteome? Whatever proteins are inside you, right? So the entire set of proteins, entire set of proteins expressed by a given cell, a tissue or organism. This is proteome. Here see, you can, if you have to study the proteome of a cell, you will consider that cell as a system, biological system. If you have to study in an organism, you have to consider that organism is a biological system. Tissue is a biological system, right? We have to apply systems biology here. Okay, now what is proteomics? Study of proteome. That's it. Proteomics is study of proteome. So simply you can say study of proteome, or you can say the entire set of, study of entire set of proteins, right, expressed by an organism. Okay, or a cell, or a tissue. Clear? Now you know it is 2026, right? The era has changed. Different technologies has came which have made the study of different things very easy as compared to earlier. Okay, so we will see main three different research developments. Okay, so three important research developments. Three important research developments, right, that have made the genomics, proteomics, these approaches possible, right? So one is high throughput technology, high throughput technology. Other will be bioinformatics, and third one will be formation of interdisciplinary research teams. Formation of interdisciplinary research teams. Okay. Now let's study the first thing. High throughput screening. Technology. Okay. Here, what we will study? This is the technology that made genomics, proteomics, epigenomics, transcriptomics, drug discovery, these things very easy. If I say genomics, this is the study of genome. Proteomics, study of proteome. Just like this, there is transcriptomics. Now what is transcriptome? You know, the study of entire set of DNA is genome, and it studies genomics. From the DNA, what you get during transcription? RNA. So whatever RNA is present inside a given organism, that total RNA is known as transcriptome. Total RNA, whatever RNA is inside an organism, full that total RNA, and it studies transcriptomics. From that, okay, here it's not just messenger RNA from which protein is made. Whatever RNA, coding, non-coding RNA, whatever, total RNA inside an organism, that is transcriptome. Okay, from messenger RNA you get proteins, right, when translation is done. So the study of the entire proteins is proteomics. Then there is, suppose there are some structural and chemical changes inside DNA, right? Because of some environmental factors, etc. And the study of those things is known as epigenomics. See, you don't need to know what is transcriptomics, epigenomics. I'm simply telling you to tell you the use of high throughput technology. Epigenomics, in which whatever chemical or structural modifications are there in chemical or structural modifications in DNA, that you study. So how is how these things are possible? These days we use computers, robots, right? We use robotics, we use artificial intelligence, we use AI, and these two things are there, and we say we are using high throughput technology. So in high throughput technology we use robotics and artificial intelligence for studying genomics, proteomics, right? For studying the genome, that is study is genomics. For studying proteome, for studying transcriptome, right? For epigenomics, and even for drug discovery. See, if I say scientists have to check to discover a new drug that can kill a particular bacteria that causes infection inside a human cell. Suppose this take out cell from your body, right? And then in cell that virus or bacteria, whatever is there, suppose we say bacteria, and we have to check antibacterial drug. Now since bacteria is there, and then the drug is injected in the cell, not inside your body, right? All these things are going on in laboratory inside a cell that has already been extracted. Okay, now this so they will take multiple cells, and multiple cells they will insert this bacteria, and then the new medicine, right now it's not medicine, right? That new molecule that if works against this bacteria, that can be a potential drug or medicine. So at same time multiple molecules can be studied in multiple cells, and that is possible through the high throughput technology. If I say human genome project, human genome project, it took 13 years, because that time the technology was not that advanced, it did not use the high throughput methods. But these days the technology is advanced. Okay, this human genome project, it was not an easy task, but these days you can sequence the DNA or RNA in a very small time, and that is possible because of high throughput technology, in which we do high throughput sequencing. Multiple genetic strands can be studied. The sequence of nucleotides can be studied at the same time because of the robotics and AI. Okay, so high throughput techniques are there for drug discovery, high throughput screening, for DNA or RNA sequence of sequence of nucleotides in DNA, RNA. The technique is high throughput sequencing, or even we say next generation sequencing. Okay, and the NGS, or this is even massively parallel sequencing. Lot of genetic strands can be sequenced at the same time. Okay, now we got lot of data from these high throughput methods. Now we need to store the data and analyze the data, and for that what is the next thing? Bioinformatics. Informatics, in which computer sciences, okay, so So in informatics, study of computational systems, you apply to biology. This is bioinformatics. So use of computational tools to store, organize, and analyze the huge volume of data that results from high throughput methods. From high throughput methods. Okay, now the third thing is formation of interdisciplinary teams. See, if some research is going on, formation of, if you have to study the entire sequence, you don't just need biologists, right? Multiple experts are needed. Interdisciplinary research teams, right? Different research team, different members will be there in the research team. So there will be computer scientists, there will be computer scientists. You are using computers, right? So computer scientists. There will be physicists, there will be mathematicians, definitely there will be biologists, there will be chemists. Okay, there will be engineers. So all this entire team will be involved in study of different things. For example, if I say whatever proteins are there inside a cell, what they are doing. So if you have to study genomics, proteomics. So in all of that you need the entire team. You can see the logo of this Human Genome Project here. Okay, this is the logo of Human Genome Project. Human Genome Project, in which you can see biology, physics, ethics, informatics, engineering, chemistry, everything is involved, right? Multiple fields are involved. Okay, my home also, formation of my home is also an interdisciplinary team. My husband, physicist, not physicist, actually is an engineer, but he is teaching physics for JEE and NEET. My father, okay, computers, right? My brothers, engineers. My father-in-law, mathematician. Okay, thank you, bye bye.